The last topics that we're going to deal with this semester are surrounding evolution. This is often divided into multiple chapters depending on what kind of book that you're reading. So it might be divided like it is here in 13 being the theory of evolution, how populations evolve, and specific ways that evolution occurs. Another chapter might define what species are and how species occur. I find that it's really hard to talk about species, populations, and evolution without touching on all of these topics intermingled. So that's what we're going to do. So instead, what we're going to do is think about it in 4 different major questions. What is the theory of evolution and what evidence is there to support it? What is a species? How do populations change over time? What is selection and what kinds of selection are there? And where do new species come from? We're going to start by reviewing some material that we talked about way back to the very beginning of the semester. And that's the theory of evolution. Books often like to call it Darwin's theory of evolution, but you'll see pretty clearly that Darwin wasn't the only person who came up with these ideas. Before we go too far, let's go back to this idea about the difference between laws and theories. So when we say something is a law in biology, and we say something is a theory, how are those two things different? Students often like to think that laws are something that are concrete, they're absolutely true, and that theories are something that are just an idea. But the real difference is what these things are meant to do. In general, laws are going to describe a phenomenon that you can observe right now, while theories seek to explain why something occurs. Both are typically well-established concepts in science, but they can change as information increases. So, how did the theory of evolution develop, and what is the support for it? Since to be a theory, there must be something. The first written documentation of a philosopher thinking about human beings and what they were before they were perhaps human was Anaximander back in 610 BC. In the beginning, man was born from creatures of a different kind because other creatures are soon self-supporting, but man alone needs prolonged nursing. For this reason, he would not have survived if this has been his original form. Anaximander pondered over the idea that in order for human babies to make it to adulthood, there must have been adults to care for them. So where did those adults come from in the first place? In addition to his philosophy, Anaximander was interested in looking at development. In particular, he spent a lot of time looking at embryos. In this image, you can see embryos from multiple different animals: salamanders, humans, rabbits, chickens, and fish. From this picture, you can see that they're actually dramatically similar to one another. And because of this similarity, he came up with this idea that all life began in the ocean, because the simplest organism that everything else looked like was a fish. On the surface, this seems like a pretty good idea, except, Anaximander was a little bit crazy. He thought that we actually were fish, like super old fish that shed our skins. Okay, probably not. He did have this idea that we maybe started out as something else, and that all organisms were related by these similarities. Another famous research scientist was Lamarck. Lamarck had this idea that creatures evolved from low to high complexity. He considered organisms like worms and insects to be low complexity and animals like mammals to be high complexity. That's pretty arbitrary, but we could kind of see where that comes from. He thought that the use or disuse of a structure might cause them to change. So if an organism used to fly but doesn't fly anymore, it might lose access to its wings. And that adaptation to the environment causes change. So anytime something is changing in the environment, we might expect the organism itself to change. These ideas don't seem that bad until you look at them a little bit closer. Lamarck frequently gets laughed at because in his version of evolution, whether or not an organism uses a trait is what determines how the trait changes. So if you're a hungry giraffe and you can't reach the leaves, if you stretch your neck long enough, eventually your neck is going to get longer, and you're going to pass on that longer neck to your offspring. Now, I don't know about you, but I've wanted to get better at lots of things in my life, and practice makes perfect, but it certainly doesn't make a longer neck. And I can't say that my kid is particularly better at any of those things I practiced. So, in general, this hasn't been proven to be very true. A lot of other people have thought about this idea of the origin of species over time. All of them agree that things are in response to a changed environment, and that this might be because of changed needs, changed behaviors, use or disuse, maybe something you learn, could be shock, it could be some kind of assimilation. And quite a few of them agree that this somehow led to natural selection, the idea that some individuals were more fit than others. What Darwin actually contributed to the theory of evolution was the idea that there was already variation that existed in nature, and that's what natural selection actually acted upon. It wasn't things that people learned per se, or a traumatic experience, or wanting to be really tall, but some individuals just happened to have some kind of trait, and that made them advantageous at that time. So this is really what he contributed, and it's a super important aspect of the theory of evolution. So how did we get there? Well, a lot of this information was originally thought about on the Galapagos Islands. These are islands that are part of Ecuador in South America. If you look at them really closely, you can see in this image that they have very different environments. Some of the islands are pretty low and don't have very many mountains or hills, and others are quite a few mountains. The Galapagos are particularly famous for the diverse amount of life that lives there. Things like Darwin's finches, saltwater iguanas, the Sally Lightfoot crab, blue-footed boobies, I don't know what that red-breasted bird is there, and, well, of course, giant tortoises. Darwin's often thought of as the most famous tourist that's been to the Galapagos, but he didn't look like this when he went there. He actually went when he was a really young man. Darwin was a huge nerd, and he was really bored at home, and his favorite thing to do was to collect animals. And by collect animals, I mean, like, collect them. Like, there's a great story in the book where he writes about wanting to collect some insects, and he had two, and they were brand new, and he didn't want to put one down, and then he saw a third, so he ended up putting one in his mouth so that he could get them all back to the ship. Like, this guy was serious. While he was on the island, he noticed that there were a large number of finches, more than he had seen in any other location, and that they were very specific to the types of foods that they seemed to eat. Some of them had very thick bills for crushing seeds, some had very thin bills for eating insects, and others had very sharp bills for cutting things like cactus flowers. All in all, there were 15 different types of finches, and each one with a specialized environment. Darwin also noticed that the birds lived in very specific places. For example, birds like the mangrove finch and the medium tree finch only lived on one of the islands of the Galapagos, while other birds like the small ground finch appeared to live everywhere. Darwin wondered how so many different versions of this bird could possibly be in one place. He started with this idea that the species likely came from South America somehow, and that that's how they all originally got to the island. Now, the bird that you see on the right here, Tiaris obscurus, is the actual descendant of all of Darwin's finches. This has been confirmed by genetic analysis, so he wasn't too far off. This meant that all of the birds were connected by common ancestry. He also thought about, why are all these birds so different from one another? Some kind of pressure in their environment had to be applied. Generally, when we think about pressure, it falls into three major categories. Either there's predators on the island that some of them need to be able to escape from, there might be some sexual conflict between members in a species, or there might be issues with food availability. Now, there really aren't any predators on the Galapagos, and finches don't really have sexual conflict, so he hypothesized that this was related to food. The idea was that some individuals must have had traits that helped them survive in one way or another. These individuals would then be more likely to reproduce and thus pass those traits on. An example of this is shown in this comic over here by Prolific Pen Comics. Two birds are sitting thinking about how they're going to get access to a grub down at the bottom. One of the birds tries, but he can't reach, and another bird shows up with a different sized beak. They make fun of him for it because he looks different from them, but it's easy for him to get the food, and they're surprised. This bird is going to have an advantage. He's got to eat the grub, so he's going to be healthier and have more energy to expend on finding a mate. In order for Darwin's hypothesis to be true, we have to first have some source of variation. So let's look at the populations that are found on the island. Here we have two species of bird, Fuliginosa and Fortis. On Los Hermanos and Daphne Major, the birds live in isolation. They don't have any competition with one another, and they can eat all of the food that is available. It's advantageous here to have an intermediate beak size so that you can have access to all types of food, but there's still some variation in how big that beak happens to be. When the two birds live together on the same island like they do in San Cristobal and Flaviana Islands, we see then that the beaks become specialized. Fuliginosa is better at eating small foods with its small beak, and Fortis becomes better at eating big foods with its big beak. So when competition is applied, the small beaks get smaller and the big beaks get bigger. But the original variation was already there. Over time, if a trait is adaptive, it will become more common in the population, because individuals who have it will be more likely to have offspring and pass that trait on. This process is called natural selection. Darwin's process requires two major observations and draws two inferences. The first observation is that heritable variation in individuals has to exist. Something about the individuals must be different, and that difference must be able to be passed on to offspring. The second is that overproduction of offspring must occur. This means that not all offspring have an equal opportunity to survive. From here, we can draw two inferences. The first is that individuals who are well suited to the environment will tend to have more offspring, and those offspring are more likely to survive, and that over time, favorable traits will accumulate in the population. It's important to stress here that individuals do not evolve. Evolution occurs at the population level. Just because you need to have a big beak doesn't mean that you will develop one, but the population as a whole will develop one over time due to natural selection, because individuals with smaller beaks will die out. This is the point where some students will say, okay, great, but I'm not so sure that the environment can really have that much of an effect on organisms and populations. So let's look at some real data. In the chart to the left, you can see the number of birds that have this size beak in millimeters. The data was collected in 1976, and the average beak size was 8.8 millimeters in width. It might not seem like a big difference, but the birds in the pictures on the right have beaks at the two extremes, with the bird on the left having a 7 millimeter beak and the bird on the right having an 11 millimeter beak. This is an obvious difference. For an animal this small, several millimeters is quite a bit. After this data was taken, there was a drought in the Galapagos. As you might imagine, droughts impact plants that are available in the area, and that means that food availability is going to change. Which plants are affected and how is going to depend on their root structure. In this image, you can see that plants have dramatically different root structures. These are all plants that are sometimes grown in prairies, except for Kentucky bluegrass, the plant all the way to the right here. If you ever wondered why it's so hard to keep a nice green lawn, it's because grass has extremely shallow roots and relies on constant rainfall to stay healthy. That means that it is not at all drought resistant, and it's going to die pretty fast when a drought happens. So larger plants with deeper roots, like trees and shrubs, are going to do better over time. There's a lot of variability in seeds too. Larger seeds tend to come from bigger plants, and these bigger plants also tend to be more drought resistant. So things like nuts from trees are more likely to be available during a drought, whereas seeds from things like grasses might not be. This means that during a drought, birds with larger beaks tend to have an advantage because they can break open those bigger seeds from deeper rooted plants. And that's exactly what the scientists saw when they went back and measured when the drought was over in 1978. The average beak size had increased by an entire millimeter. Now that's something interesting to say if it happened only one time, but it actually happens every time a dry year happens. So if we look at beak size over time at the Galapagos Islands, we can see that for this bird, we see an increase in beak size every time there's a dry year. Once in 1977, again in 1980, again in 1982. And in fact, we can follow this like clockwork. So even a year's worth of drought is enough to cause noticeable change in a bird. So why doesn't this increase in beak size persist? Well, in wet years, there are tons of seeds that the smaller beaked birds have an advantage at eating. So we actually want to keep small beaks in case there's a really wet season. Permanent changes are difficult to get, and we'll talk a little bit more about them when we get to speciation. Suffice it to say, there are enough differences in the foods that are available and the types of environments on each of these islands that a lot of specialized birds have since evolved. And that's where species comes from. If you have these small effects and they happen long enough, then you can form a new species over time. So the key ideas here are that all life shares a common ancestor. Natural selection acts on natural variation in a population. This is variation that already existed. Individuals with beneficial traits are more likely to pass those traits on to the next generation. And over time, those traits can become more common. If this happens long enough, eventually it can lead to speciation. There are a lot of different myths that I hear about evolution when I'm teaching courses like this. So I'm going to go over a couple of them. The first one is that the theory of evolution says that humans evolved from monkeys. That's not actually true. The theory of evolution says that humans and monkeys share a common ancestor. And common ancestors are species that were neither of the current ones. So that means that our common ancestor was neither a monkey nor a human. I also sometimes hear that you can't believe in God and evolution at the same time. That's not true either. Evolution makes no comments on whether a deity exists or where life ultimately came from. It only seeks to explain why and how populations change over time. That's of course not true if you're talking about young Earth creationists who reject things like fossil records, and literal creationists are the only exceptions. But in general, it doesn't say those things, and it's not a problem for people who are devout. In fact, Darwin himself was very religious. It's also not really something you can believe in or not. It's a theory. You either agree that there's support for it, or you don't. So moving on to that, let's talk about support for the theory of evolution, and where it comes from. We're going to talk about 4 major types of support. The first one is artificial selection, the second homology, both structural and molecular, the third fossil record, and the last physical observation. Artificial selection is probably the one that you're the most familiar with. Artificial selection differs from natural selection in 2 major ways. The first is that the selection pressure comes from the desires of another organism. Most of the time, that's going to be humans. So humans decide what traits they consider valuable, and they select for those traits. And the second is that these favored traits are usually not based on survival and reproduction, which is not the case for natural selection. An example of this would be how we select crops. For example, natural corn used to be extremely small, and even though it was edible, it wasn't particularly efficient to grow. However, humans have selected for different varieties of corn that are capable of growing in different environments, produce a lot more sugar, and are much larger. Other examples include things like the brassica family, which produces cabbage, Brussels sprouts, kohlrabi, kale, broccoli, and cauliflower. Each of these vegetables was selected based off the desire of individuals to eat particular parts, be that terminal buds, lateral buds, stem, leaves, stems and flowers, or flowers themselves. You can also find traces of artificial selection in collected animals, like pigeons, for example, which are often bred to create fanciful varieties that would have a difficult time surviving in nature. Artificial selection shows that all of these traits are heritable, and if individuals are selected to breed with those traits become much more common in the population. So they support all of the underlying bases of natural selection. The next thing that we'll talk about is homology. Homology is the state of having the same or similar relation, relative position, or structure. And we call things that have homology homologs. And generally, these things share a common ancestry. There are 2 major types of homology. One of those is structural homology, and the other is molecular. Structural homology has to do with the way things are structured, pretty much what it sounds like. For example, if we were to look at the forelimbs of a human, a cat, a whale, and a bat, we would see that the types of bones involved and the Hox genes that control the production of those bones are highly conserved. These are called homologous structures. They all have different functions, but they're located in the same locations in the animals, and they are made up of the same basic structures. We also see homology when we look back at the embryos that Anaximander was so infatuated with. It explains why early stages of development in different animals are very similar, even though those similarities might not be visible in organisms when they're adults. For example, both chick embryos and human embryos have pharyngeal pouches, which will eventually become gills in things like fish, and they have a post-anal tail. The tail will shorten and be reabsorbed in human embryos most of the time. These are considered vestigial structures, which means that they're remnants of features that were important in the organism's ancestor, but are no longer important for the organism's development. Sometimes these vestigial structures are not reabsorbed properly, and in the case of humans, the most common case of that is the growth of a post-anal tail. These are normally surgically removed before a kid is 5 years old to make it easier for them to sit and develop properly. Other examples of vestigial structures are the presence of both a femur and a pelvis in whales, which is an indication that their ancestors once walked on land, as well as limb structures like coccyx and pelvic girdles in snakes that suggest that their previous ancestors also had limbs. Usually the only visible remnant of this is going to be a spur that is visible right near the base of the tail. Sometimes students conflate the idea of analogous and homologous structures. So we've been talking about homologous structures. These are structures that have similar placement and similar structure to each other. Analogous traits have similar functions but have completely different structures, and they tend not to come from common ancestors. So examples of homologous structures are things like the forelimb in all of these animals: humans, horses, cats, porpoises, and bats. They all use the same combination of bones and are regulated by the same Hox genes. Analogous structures would be things like wings. The wings of a moth, a bat, a bird, and a pterodactyl are going to have very different structures. They use different parts of the body to create lift, and they do not have a common ancestor as a result. So remember, homologous structures are not the same thing as analogous structures. Homologous structures have similar structure and placement, and they share a common ancestor, whereas analogous structures have similar functions but different structures, and they do not come from a common ancestor. Not all homologies are obvious because they're not all physical structures. Some of them are molecular. For example, the fact that Hox genes are conserved across organisms, from a fly to you, means that they are homologous. They have similar structures and similar functions. Another example of this is how similar specific genes are. In this image, you can see the sequence for the mouse gene PAX6. PAX6 is important for eye development and eye control. The sequence similarity for all of these, from the fly, shark, squid, and flatworm, are pretty similar, but the proteins are identical. So not only does this gene perform the same function in controlling the development of the eye, it also produces the exact same protein. Let me give you another example. We'll talk about a disease that affects some individuals. It's called Parkinson's. Parkinson's is usually associated with a tremor, but it actually causes a lot of other symptoms. Things like problems with balance, increased stress and anxiety, thinking and memory problems, and sleep problems. This is caused by a specific gene called synuclein alpha, or SNCA. Mutations in this are autosomal dominant, and they cause Parkinson's, and sometimes they're implicated in Alzheimer's and Lewy body dementia. Here's an example of the sequence of this gene, SNCA, between humans, mice, cats, dolphins, and elephants. Notice that the vast majority of this sequence is identical. All of these organisms are capable of having Parkinson's just like a human being. So even though you haven't seen an animal that has this disease, doesn't mean that it doesn't happen. Molecular homology and structural homology are a big basis of biomedical research. If animals weren't similar enough to human beings, we wouldn't be able to use them to learn information about ourselves. So you can thank this molecular homology and our relationships evolutionarily for all of the vaccines and other medicines that we've been able to test using animal friends and animal DNA sequences over the lifetime of genetics. Sometimes we don't have the luxury of being able to look at an animal that's alive right now or getting a DNA sequence. When that's the case, we can turn to fossils. Fossils are the imprints of remains of organisms that lived a long time ago, and they document differences between past and present organisms and can sometimes reveal the many species that have become extinct before now. In some cases, the fossil record also reveals historical sequence in which organisms have evolved. Part of what makes the fossil record so powerful is that it is essentially a timeline of events. As each generation happens, the next one occurs above it, and so the farther you dig backwards, the farther back in time you can go. Of course, to analyze them, biologists need help from geologists and paleontologists, so this isn't something that we tend to do alone. Many fossils can link early extinct species with species that are living today, and when new fossils are found, we can use those to rearrange how we think species are related. An example for that is how we know about the origin of whales. Beginning in the late 1970s, paleontologists unearthed transitional fossils that were thought to come from whale-related carnivores. These creatures look a lot like wolves, and they thought they were related to whales mostly because of their tooth structure and their skull structure. However, molecular biologists found close relationships between whales and hippopotamuses instead of wolves, and they hypothesized that whales and hippos were both descendants of the same cloven-hoofed ancestor. It's not possible to go back in time, so how can we know who's right? One thing that we can use is the structure of bones and how they are formed within the fossil record. For example, we know that there are two different types of hooved animals. There are perissodactyls, which have odd-numbered toes, like the tapir, rhino, and horse, and there are even-toed hooves, like cloven hooves, which occur in pig, deer, camels, hippos, and pronghorns. These things have specific structures. For example, if we were to look at the knuckle bone, we can see that in odd-toed hooves, there are two humps at the top and we're missing the spur at the bottom. But in even-toed hooves, we can see that there's a double bump at the top, but there's also a connective spur at the bottom. Because whales and hippopotami are closely related molecularly, we would expect the ancestor of whales to have a cloven-toed ankle. So did the wolf-like organism have an ankle that looked like this? No. Instead, it has an ankle that's consistent with having an odd number of toes. So even though we don't have access to its genetics, we can say that it's not closely related to an hippopotamus, so it's probably not closely related to a whale. What about the other organism that might have been related? If we look at its ankle bone, Pachycetus, we can see that it's actually very similar to what we see in pigs and deer, which are cloven-toed organisms. This suggests that it's more closely related to the hippopotamus and more likely to be an ancestor to whales. This allows us to create a better timeline and include the correct organisms when we think about how whales evolved from land-dwelling animals to living in the sea. Do I expect you to remember all these details about whales? No, of course not. But what I'd like for you to get out of this information is that we can use the fossil record to try and give us information about organisms that we can't otherwise study. And we can use that to compare to organisms that are currently alive and see whether or not what the fossil record suggests relates to what we can see in the world today. In addition to that, we can observe things directly. There are lots of ways to document evolutionary change. For example, I already gave you information about how we can watch birds change in the Galapagos based on the amount of rain. But two common things that we talk about day to day is resistance to things like antibiotics and bacteria, and pesticide resistance in insect species. This is different from artificial selection. Artificial selection is selecting for a trait that has nothing to do with survival, but is something you want to happen. Under most circumstances, when you apply pesticides to, let's say, a crop field, maybe by crop dusting like in this plane, you're trying to kill everything. It's essentially a genocide on this insect. This is an extremely heavy evolutionary pressure. This means that only a few organisms are going to survive, those that are resistant to your pesticide. They are the only ones that are going to be able to have offspring, and that means that you're going to increase pesticide resistance every time you spray the field. This is the same thing that happens when people overuse antibiotics. If you don't kill everything and you leave some survivors behind, those resistant individuals are the only ones that can produce offspring, so the next round of antibiotics won't work. Remember, this is not an example of artificial selection. This is natural selection. Natural selection doesn't mean that humans aren't involved. After all, human beings are part of nature too. All of these examples of evolutionary adaptation reveal two important points about natural selection. The first is that natural selection is more of an editing process than a creative mechanism. This means that it doesn't just create spontaneous things. Instead, it makes small changes to what is already existent. The second is that natural selection is contingent on time and place. It favors heritable traits in a varying population that fit the current environment. That means that something might have been advantageous a few years ago that might not be advantageous today, but you're still going to see it. It's important to remember that natural selection does not create perfect organisms. It's only going to help produce organisms that are better at living in the current environment. Alright, now that we've talked about what evolution is and some support for it, let's talk about what a species is, so that we can think about how new species occur. Usually when I ask students what does species mean, they tell me that it's a group of organisms that can interbreed and produce fertile offspring. And that's mostly true. But that's only one definition of what a species can be. Although the basic idea of species as distinct life forms seems kind of intuitive, defining a formal definition is actually pretty difficult. And there are four major ways that we can try and define a species. And I'm going to talk about why they're useful and where they fall apart. Those four ways are the biological species model, the morphological species model, the ecological species model, and the phylogenetic species model. The biological species concept, or biological species model, is the one that you're familiar with. This is the idea that a species is a group of populations whose members can interbreed and produce fertile offspring with each other, but not with members of other species. This concept emphasizes something called reproductive isolation. The classic example of this is the mule. For example, horses and donkeys are able to mate, but they have different chromosome numbers. This means that when they mate, they produce an organism, a mule, that has an odd number of chromosomes. These chromosomes cannot pair properly during meiosis, so the mule is sterile. This is used to emphasize the fact that horses and donkeys are different species. However, that's not always the case. Sometimes hybrids are not sterile. One example of that is thinking about wolves. The three canids that you can see in this picture are C. lupus, the gray wolf, C. rufus, the red wolf, and C. latrans, the coyote. These organisms have long been thought to be separate species. Recent genetic research has shown that C. rufus appears to be genetically a hybrid between the gray wolf and the coyote. If it is a hybrid, it's a fertile hybrid, suggesting that gray wolf and the coyote are not distinct species. Anyone who's observed a gray wolf or a coyote is pretty sure that they're not the same thing. They don't look similar, they don't behave similar, and they don't usually live in the same places. C. rufus is similar. The red wolf lives in different places than the gray wolf most of the time. It has a little bit of overlap in where it lives. So, which one is correct? Is the red wolf a species? Is it a recent hybrid? Is it an ancient hybrid that somehow is now a species? Do different species of wolf that recently hybridized with coyotes count? Are they true breeding? Well, yes, but then what are they? The answer to this question is we really don't know. According to the biological species model, they're not a species, but according to what we might think intuitively, they seem like they are. There are other places where the biological species concept falls apart, and that includes anything that reproduces asexually. The biological species model requires something to only be able to breed with other members of its population. But if you don't breed with anyone, how would you know whether or not you could make a hybrid? This means that it breaks down for plants which can hybridize easily and are usually viable, for most bacteria, for some parthenogenic organisms where the female doesn't need a male, like fish, insects, and reptiles, and of course things that are fossils, because who knew who they could mate with? So even though the biological species model is the one that we're most familiar with, it doesn't really work for everything, and there are some serious problems with it. The morphological species model, or morphological species concept, is based on observable physical traits, and is usually applied to asexual organisms and fossils. This is helpful because we can't really see those specific organisms mate with each other. An example would be how we looked at whales before. We can use things like the structure of their ankle to determine whether or not they're related. But the morphological species concept also has problems. For example, sometimes species look so similar to one another that by morphology we would probably not be able to tell them apart. In this example, you can see both an eastern and western meadowlark. The morphological species concept would consider them a single species, but they actually cannot produce viable offspring with each other, so the biological species concept would say that they are distinct species. So when things look too similar, sometimes the morphological species concept breaks down. The ecological species concept, or species model, defines species by where they live and the ecological niche that they occupy. This is helpful for separating species that might live close to each other but don't actually have overlapping territories. For example, in the image here, we're looking at a lot of different salamanders that are somewhat similar to each other. However, they live in different locations, their territory does not overlap, and the things that they eat is also very dissimilar. So they're actually different species. The phylogenetic species concept, or species model, defines species as the smallest group of individuals that share a common ancestor. It usually uses genetics or sequencing to try and relate species. So things that are extremely similar to each other are more closely related to things that are particularly different. Normally, these organisms will then be organized into phylogenetic trees, which are based off of how closely related they are. So for example, in this tree that you're looking at here, Canis latrans and Canis lupus are considered separate species based on their genetics, but are closer related than, say, Canis lupus and Lutra lutra, the otter. So now that we have all these species concepts, how do we actually define a species? Well, generally, we just try to apply species consistently, but it's really, really hard to do. It's pretty controversial sometimes, if something is a species or not. And then there's the idea of subspecies, and that's controversial too. So, in general, we just try to use as many of the concepts at once as we can. The more that fit, the more confident we can be that two things are different species. Alright, so with all that said, most of the time we'll still be relying on the biological species concept, because it's the most common one that we like to use. I mentioned that it relies on an idea of reproductive isolation. Reproductive isolation is set up by what we call reproductive barriers. These are things that keep species separate. They serve to isolate the gene pool of species and prevent interbreeding. Most reproductive barriers can be divided into one of two things. Either they're prezygotic, which means they happen before the zygote forms, or they're postzygotic, which means that they happen after. So let's go through some examples of each. Perhaps the most obvious example of prezygotic separation is just being in different places. Habitat isolation means that these organisms never encounter each other, so they don't have the opportunity to interbreed. An example of this is the garter snakes that are pictured here. Thamnomorphus atratus lives mainly in the water, whereas Thamnomorphus sertalis lives on land. Because they live in completely different locations, they don't have to worry about having to interbreed or not. Another example is temporal isolation. This happens when two organisms are not ready to breed at the same time. For example, these two skunk species have overlapping territory, but one of the skunks goes into estrus in late winter and the other one is ready to breed in the fall. If a male from one species encounters a female from the other, they won't be interested in each other at the same time. This is a classic example of just not being in the mood. Another example is behavioral isolation. This happens when different organisms have different courtship rituals. Examples are things like blue-footed boobies and masked boobies. The blue-footed booby does a dance involving its feet, and the masked booby does a dance involving its bill. If the male does the wrong dance for the wrong female, she won't be interested in him at all. This is very similar to human courtship when we think about it. Everybody likes what they like, and you have to know the right dance if you want to catch the girl or boy. Another form of separation is mechanical isolation. This happens when, well, things just don't really fit. And let's say we get all the way down to the end. We live in the same place, we're ready at the same time, we like the same things, and everything fits fine. But things just don't work out. That can be gametic isolation. This is when there's an incompatibility between eggs and sperm. And this is something that's very common for things like broadcast spawners, like these sea urchins here. Sea urchins really spawn just by kind of throwing it all out there. They hurl all their eggs and all their sperm into the ocean, and then they hope for the best. Since there are multiple types of sea urchins using the same strategy, they need to make sure that their eggs and sperm are only compatible with the eggs and sperm from their own species. And that's gametic isolation. These are all of the major ways that prezygotic barriers can work. You can either not live in the same place, in habitat isolation. You cannot be ready at the same time, that's temporal isolation. You could have different courtship rituals, behavioral isolation. Things could just not mechanically work, mechanical isolation. Or your eggs and sperm may be unable to fuse with those of another species, and that's gametic isolation. So what if all of these things fail, and you actually produce a zygote? Well, then you move to postzygotic barriers. The most common and perhaps most obvious postzygotic barrier is that, well, maybe the hybrids that you produce are just really crappy. Either they die very early or they don't really get born at all. That's reduced hybrid vitality. You could have a situation where your hybrid is infertile. That's like the mule example we talked about before. This produces sterile hybrids, which prevents the species from really interacting for long term. The other option is that you could have hybrid breakdown. This happens when fertile offspring are created, but those fertile offspring are unable to have fertile offspring of their own. And this is pretty common for plants. Now we've talked about what a species is, or rather how difficult it is for species to be defined, and some of the things that keep species separate. So now we're going to spend some time talking about the evolution of populations. Before we can talk about how populations change, we first have to spend some time talking about variation. Organisms typically show individual variation, and mutations are the ultimate source of this genetic variation. They're where alleles come from originally. Assortments of existing alleles can arise in a variety of ways. We can have things like crossing over, mix the alleles around. We can rely on independent orientation of homologous chromosomes in metaphase 1. And of course, we can also look to random fertilization. All of these things are going to work together to create the variation that we see within a population. So what is a population? A population is a group of individuals of the same species that live in the same area and can interbreed. When we talk about populations, we also sometimes talk about this idea of the gene pool. A gene pool consists of all copies of every type of allele at every locus in all the members of the population, so all of the possible things that anyone might inherit. And we also connect this to a term called microevolution, which is a change in the frequency of alleles in a population's gene pool and the evolution that occurs on the smallest scale. When we think of evolution, we often limit this idea to natural selection, but there are actually three main causes of evolutionary change. Natural selection is only one of them. Genetic drift and gene flow are the others. There are also multiple types of natural selection: stabilizing, directional, and disruptive. So let's talk about how these might differ from each other. Natural selection can occur in three alternate ways. Let's say that we're starting with a population of mice. These mice can come in colors from white to black, but most of the mice are somewhere in the middle. Depending on the type of pressures that are applied to this, we might see differences in how this population will change over time. One example of this is stabilizing selection. If the extremes are disadvantageous, being white or being black, then we will see an increase in the intermediate color in the middle. This reduces variation and makes the species more consistent. If instead one of the extremes is favored, we might see a shift in where the average color is. Say, for example, mice that are white are at a disadvantage in this environment. Maybe the advantage is lots of dark grays or browns. We might see a move away from the white mouse and toward the darker colors. This is called directional selection. It doesn't reduce variation, but it changes where the peak in that variation occurs. Now let's say instead of having the middle be advantageous, the two ends are advantageous. Perhaps it's better to be either white or black, and anything in between is bad. This results in something called disruptive selection, where the two extremes end up being chosen. This can result in two different morphs or versions of a species without any intermediates. So let's talk about some real-world examples. One example of directional selection is things like weight. Larger animals have an easier time keeping warm in the winter, and smaller animals have a harder time regulating their temperature. When there is a cold snap, this selects for higher weight birds. What you're looking at in the graph here is the original population, which has quite a bit of variance in body size. After cold weather occurs, only the larger birds are left. This results in a shift in the average size of these birds, and it is an example of directional selection. Now let's think of an example of stabilizing selection. Have you ever thought about why most human babies are somewhere around 7 pounds? What is it exactly that makes this an ideal weight? Well, it has to do with survival. The smaller a baby is, the less likely it is to survive after birth. And the larger a baby is, the more likely it is to get stuck in the birth canal and result in complications for both mother and child. This is particularly bad in areas where medical interventions like C-sections are not available. This means that there's an ideal size for babies if they want to protect both their own survival and the survival of their mother, and that's around 7 pounds, the ideal weight and size to be pushed through the birth canal with no complications. An example of disruptive selection has to do with beak size. So, for example, you might have a situation where high variability in beak size is advantageous because there are many types of food. However, after a drought or on specific situations, there might be fewer types of food, and so specific extremes will allow birds to specialize on the type of food that they can eat, separating into two different morphs, one that has a large beak and can eat larger, more difficult foods, and one that has a small beak and can eat smaller and more dex-requiring foods. This is very similar to what we talked about for Darwin's finches, where different food requirements and pressures for food consumption resulted in disruptive selection for beak size that eventually led to speciation. Another form of natural selection can be sexual selection. In this form, individuals with certain characteristics are more likely than others to obtain mates, and this usually involves secondary sex characteristics that gives them some kind of advantage. Good examples of this are when you see species where the male looks different from the female. This is called sexual dimorphism. For example, in peacocks, the size of the male's tail is an indirect measurement of how healthy he is. It basically says to the female, look, I can waste resources on this huge, beautiful tail and still escape predators. I'm extremely fit. You should pick me. Another example is things like antlers in deer. These can be used also to indicate how healthy the male is and to make him better at fighting. One thing to consider about all of these processes is that natural selection is the only form of selection that generally leads to adaptive evolution. Let's use an example that's similar to something you did way back at the beginning of class. In this example, we have one species, and that species is variable in what color it is. It's either red, purple, yellow, or green. And that the selective pressure that we'll be using will be predation. Remember that natural selection is based on what is occurring right now. So where the species lives is going to have a big impact on whether or not any of these colors is advantageous. For example, if we have the Teenage Mutant Ninja Turtle environment that's listed here, what would you expect to happen? Based on the colors in the environment, we might expect that green would be favored, and most of the time that would be true. This is real data from 2 previous classes that shows that in each case, green was favored. At one point, so much so that every individual that lived in the environment eventually became green. Now let's think about a different environment. In the sunflower environment here, we might expect yellow and green to be favored over red and purple. However, that's not always the case. Here's some data from previous students doing this experiment. They found that in one case, yellow was heavily favored to the point of all individuals being yellow, but in other cases, other colors were equally likely to be around. In this case, in the second group, red has the most favor, and green and yellow are bouncing back and forth. So how is this possible? Well, let's think about two scenarios. In the first scenario, individuals in this population fall into these places. Individuals that are red and purple happen to stand out quite dramatically. However, if instead the red individuals happen to fall where the sunflower cores are, they might have a better chance of hiding. So where individuals are in their environment when a predator reaches them is going to determine whether or not their color is advantageous. This introduces quite a bit of chance into this scenario. Let's think about another example. If we look at the Spider-Man environment here, we might expect that purple and red are going to be advantageous. And in fact, we can find that all three colors under different circumstances that we see here, purple, red, and yellow, have some kind of advantage, and that green is pretty consistently least advantaged. However, exactly what's going to happen again is due to a lot of chance. Why is there variability in which color is going to be advantageous? Well, a lot of it has to do with variability in predators themselves and also this idea of chance. So predators might prefer or notice prey that are closer to them. They might have an affinity for a specific type or color of prey. Maybe the predators didn't follow the directions or read them. Maybe they ate more prey than they were supposed to, and some predators might not have the ability to tell the colors apart. Individual prey might, by chance, land in areas that are a good fit for them, or not. So when we're thinking about natural selection, we often simplify everything down to, does the color match the environment or not? When there are in fact a lot of variables that are working together to determine what's going to happen. This is what we mean by natural selection does not create perfect organisms. It's only going to select for things that are advantageous right now in this instance, given all things the same. One of the things that happened during these scenarios is that in some cases only one color remained. This is called reaching fixation, and that's when variation is reduced to one phenotype. This doesn't actually happen all that often in the wild. So why not? Well, that's because diploidy actually preserves the variation by hiding recessive alleles. And sometimes things like balancing selection occur, when natural selection maintains stable frequencies of two or more phenotypes in a population. There are also situations where the heterozygous form has an advantage, which would mean you would want to keep the recessive trait around. This is a type of balancing selection where individuals that are heterozygous have greater reproductive success than either homozygote. Let's talk about an example of how this might happen. And we're going to use sickle cell anemia. Sickle cell anemia is unfortunately a disease that causes an awful lot of problems for individuals who have it. It changes the shape of normal red blood cells. Instead of the standard round shape, red blood cells are stretched out into sickles or half moons. You can see them in this picture of actual red blood cells to the right. Normally, red blood cells can easily pass through blood vessels because they're round and somewhat flexible, but sickle cell red blood cells have a difficulty doing this, and they sometimes will get stuck and cause blood clots. This means that not only are they poor at carrying oxygen and lead to anemia, they also increase the likelihood of blood clots and things like stroke and pain in individuals who have them. Sickle cell anemia is caused by a recessive mutation. So if having sickle cell anemia is so bad, why is this something that we want to keep around? Well, individuals that have sickle cell trait are resistant to malaria. Malaria is a disease that enters the red blood cell and is able to multiply inside of it. Sickle cell red blood cells do not allow this to happen, so only 50% of the red blood cells in a heterozygous individual are susceptible to malaria. This means that they get a much milder form of the disease and recover much quicker. So in areas where malaria is present, being heterozygous is a huge advantage. All right. So now that we've spent a lot of time on natural selection, let's talk about other ways that evolution can occur. One of the other things that we can have is gene flow. Gene flow is effectively changes in allele frequency due to migration between populations. So if you have one population that's full of recessive individuals and you have someone in a population from dominant individuals and that person travels, that would be gene flow. Gene flow is something that's made really easy by things like airplanes and the way that we intermix today. So we can look at how many people move from one location to the next location and whether or not that has an impact on the alleles that are present in the gene pool. The other thing that we can consider is genetic drift. This is changes in allele frequency that are mostly due to chance. They're not due to natural selection. An example of this might be you're walking down the street and you happen to step on a bunch of ants. Some of the ants are protected by the tread of your shoe. The ones that happen to survive would be all that's left to make the new population. The survivors have nothing to do with what their alleles actually are. No selection event has happened. This is genetic drift. Rocks fall, some people die, whoever's left, that's who gets to carry on. This can lead to massive changes in allele frequencies and the evolution of traits that might otherwise seem very disadvantageous. There are two big categories of genetic drift, and those are the bottleneck effect and the founder effect. The bottleneck effect is loss of genetic diversity when a population is greatly reduced. So that example of rocks fall and some of the population disappears. It's called the bottleneck effect because it's like putting a bunch of marbles inside a bottle. When you flip the bottle over, only some of the marbles will be able to fall out, and only those that do will make up the new population. Examples of bottlenecking effects can be things like poaching, or having something dumped into an ocean or river. Usually the bottleneck effect can lead in two things, either a recovery of the population with reduced variation or extinction. The founder effect is a little bit different. In this case, a few individuals will colonize a new location, like an island, and this results in low diversity. For example, let's say that we have a sample original population. Founding population A consists of 3 different options, 2 black beetles and 2 different types of spotted beetles. This means that the descendants of population A are going to be missing all of the gray beetles that were in the original population. The founding population B has gray beetles instead of black beetles. This means that there will be no black beetles in the population that they found. So even though there's high diversity in the original population, there's lower diversity in the new populations because the founders of those populations had lower diversity. Founder effects can also lead to an increase in the number of individuals that have deleterious traits, and this is one of the reasons why some groups of individuals are associated with specific genetic disorders. Alright, so now that we've talked about ways that evolution can happen, how can we tell whether or not it's occurring? One thing that we can do is use a model called the Hardy-Weinberg equilibrium. It states that allele and genotype frequencies will remain the same or constant if a population is large, mating is random, and there is no mutation, gene flow, or natural selection. i.e., if evolution isn't happening, then the gene pool shouldn't change. The equation itself is pretty simple. P squared plus 2pq plus q squared equals 1, where p is the frequency of the dominant allele and q is the frequency of the recessive allele. Essentially, the equation comes from calculating the frequency of getting any of these genotypes. Homozygous dominant would be p squared, heterozygous 2pq, and homozygous recessive q squared. Let's say, for example, we start out with a frequency of the dominant allele being 0.3 and the frequency of the Q allele being 0.7. We want to calculate what is the frequency of each of these possible genotypes. We can plug that into the Hardy-Weinberg equation. When we do that, we get that the frequency of homozygous dominant individuals should be 9%, heterozygotes should be 42%, and homozygous recessive individuals should be 49%. Let's say that instead of being given allele frequencies, we can see individuals and their traits. This time we have different types of iguanas, and they either have unwebbed claws, which have a dominant allele, or they have webbed claws, where they have 2 recessive alleles. When we look at the population, we see that there are 320 that are homozygous dominant, 160 that are heterozygous, and 20 that are homozygous recessive. We can use these numbers to calculate the frequencies of each of these genotypes. So that would be the number of individuals with that genotype over the total number of individuals in the population. We can also use these numbers to calculate allele frequencies. For example, each individual who's homozygous dominant has 2 of these alleles, so we would use twice those number of individuals, so 640 instead of 320. And then each individual who's heterozygous has 1 copy of the dominant allele, so we would add 160. That gives us 800 dominant alleles over 1,000 possible alleles, or 80%. We can use the same process to calculate the recessive alleles, or we can just realize that Percentages and ratios have to add up to 100. So if 80% are dominant alleles, 20% have to be recessive. Now that we understand how to calculate allele frequencies either from population numbers or from frequencies that I've already given you, whether or not a species is evolving is basically, are those population frequencies the same? If they were 0.8 for the dominant the first time and they're 0.8 for the dominant today, then no change has occurred. I recommend that you go and check out the practice problems that I've posted on Canvas for you. There will be at least one Hardy-Weinberg equilibrium problem that's very similar to the practice problems on your final exam. There's also a key on how to solve these problems, and if you have any questions, make sure that you make a trip to office hours. The last topic that we're going to discuss is mechanisms of speciation itself. Today's biologists represent patterns of descent using evolutionary trees. These are often turned sideways, and homologous structures can be used to determine the branching sequence of those trees, so we can use anatomical structure or molecular structure. These kinds of evolutionary trees look a lot like this, where each of the nodes, labeled 1, 2, 3, 4, 5, and 6, is a common ancestor between subsequent branches. So if all of these different branches represent species, where did the species come from in the first place? There are three major ways that species can form. One of those is allopatric speciation. This is caused by geographic isolation. Sympatric speciation happens when a new species occurs in the same geographic location as its founder species or parent species. And adaptive radiation occurs when multiple species arise from the same individual species. Let's give an example of how allopatric speciation works. Let's say that we have a scenario where there are two different environments that are close together, the sunflower environment and the Spider-Man environment. And these two environments are separated by a mountain range. A population is able to move in between these two environments and consists of 4 different colors. While they're traveling one time, a bunch of rocks fall, separating the populations on either side of the mountain. Now there is no more gene flow that can occur between the two sides. We've already talked about how these two different environments have different selection pressures. So for example, in the sunflower environment, yellow tends to be favored, whereas in the Spider-Man environment, purple or red tend to be favored. This means that over time, the populations are going to have different selective pressures, and they're going to change. Eventually, they will no longer look like each other, even though they are only separated by this tiny mountain range. This actually happens pretty commonly. An example of this is in different ground squirrels that exist near the Grand Canyon. They were originally separated when the canyon formed and are no longer able to interbreed. They now look a little bit different from each other and have completely different behaviors. And this is a good example of allopatric speciation. Sympatric speciation occurs when a new species arises within the same geographic area as its parent species. This is very common in plants because they can evolve by polyploidy, or duplication of chromosome number due to errors in cell division. Habitat differentiation and sexual selection, usually involving mate choice, can also lead to this. So let's talk about an example. Let's say that we have a habitat that looks a lot like this, and we want to think about how the habitat differs. We have 4 different colors of individuals, and in general, purple individuals are going to be favored. However, there are places where individuals in the green and yellow categories might be able to reside. If individuals in the green and yellow categories start living primarily in these locations, this could result in specific phenotypes staying in those places and eventually reproductive isolation. If that happens, this could eventually lead to speciation. There are no physical barriers between the green and yellow individuals leaving these parts of the habitat, but there are predator barriers that are keeping them from leaving, so they don't. On the plant side, parent species can sometimes go through errors in cell division. In this case, what has happened is the cell has replicated its DNA but fails to divide. It's now gone from a diploid 2N species to a tetraploid 4N species. When this species goes through cell division, it's going to create diploid gametes. Those diploid gametes are only going to be able to go through self-fertilization, which is fine because it's a plant, so we'll continue creating tetraploid plants. We actually do this on purpose, so we can treat plants with colchicine to increase the number of chromosomes that they have. This is why the varieties of plants that you see in the grocery store are so much larger than their wild varieties. We've actually increased their chromosome number to make them create more of all of the things they would normally produce. Hybridization of plants is also something that can lead to new species. For example here, species A is diploid 2n equals 4, and species B is diploid 2n equals 6. When these two hybridize, they create an individual that is n equals 5. This is a problem because chromosomes cannot pair, but it's okay because plants can replicate asexually. However, if there's an error in cell division that now makes this individual diploid again, this can create a fertile hybrid species. This is actually where things like seedless watermelons come from. So seedless watermelons are created by hybridizing a watermelon with 2 sets of chromosomes to a watermelon with 4 sets of chromosomes. This results in a watermelon that has 3 sets and is otherwise seedless. However, sometimes there can be an error in cell division that creates a seed that is perfectly fine and diploid again, making it 6N. This is why you can sometimes find seeds in your seedless watermelon. The last type of speciation that we'll talk about very briefly is adaptive radiation. This usually happens when a new area is colonized by a species that then rapidly changes. That's what happened with Darwin's finches. The first finch arrived in the new islands and then rapidly colonized all of the subsequent islands. The same thing occurred in Hawaii, where a founder species bird gave rise to all of the major species that now live there. Adaptive radiation is usually found in birds and fish, because these are species that are able to get places that other species cannot very quickly, and often get trapped in those locations. And that's it. Thank you all for such a wonderful class, and don't forget to fill out your course evaluations.